So, welcome everybody. My name is Carmel Shachar. I'm the Executive Director of the Petrie-Flom Center for Health Law Policy, Biotechnology, and Bioethics at Harvard Law School, a center whose name indicates that lawyers should never name anything because we use all the words, but also a center whose mandate is really reflected in the name. We look at innovation when it happens in medical research, in healthcare delivery, and healthcare financing, to see how that is impacted by and impacts our legal, ethical, and regulatory systems. So before I tell you a little bit about this event, I just want to say that one of the missions of the Petrie-Flom Center is definitely to strive to deliver interesting programming to the Harvard University Boston community. And so if you like this event, please consider going to our website and signing up for our newsletter. We promise we don't spam. We take it very, very seriously. It's a newsletter that comes out twice a month, and it gives you a list of other events that we're hosting or that our sister programs are hosting. So today is the culmination of Outbreak Week, which has really been the baby and brainchild of the Harvard Global Health Institute that we've been very lucky to partner with. And I think that this symposium, which is looking at preventing epidemics in a world that is increasingly connected, is so timely. Even though the epidemic that inspired it, the 1918 Spanish influenza, was 100 years ago, I think that there's still this undercurrent of anxiety in our society about what happens when the next big one hits. And you see it when Zika comes out, you see it when Ebola comes out, you even see it, and here, you'll forgive me because I am going to spoil the most recent Avengers movie. But you see it in the most popular summer blockbuster that ends with 50% of the universe's population disappearing, and the anxiety of what happens when our loved ones are all of a sudden taken away. And I think right now the answer is we don't have great answers, but there are a lot of people working on, well, what will it be like when the next one finally hits? And how do we prepare ourselves so that that experience isn't the experience of the Spanish influenza, so that it isn't the experience of the Avengers Infinity War? And again, sorry for spoiling it. This is such an important topic because without preparation, we really will be facing those scenarios. So I'm thrilled that we're bringing this conversation here to Harvard University with some immensely talented speakers. But before we get to those speakers, I want to introduce the man who has really been the driving force behind Outbreak Week to remind us to have these important conversations about preparedness for outbreaks, about what is the role of the media, about vaccines. And that man is Ashish K, who is the Dean for Global Strategy and the KT Lee Professor of Global Health at the Harvard T.H. Chan School of Public Health, as well as the Director of the Harvard Global Health Institute. So with that, help me welcome Ashish K.
Let me turn it over now to our keynote speaker, Tony Fauci, who in this room needs no introduction, but I will say only this. There have been many ways that I've heard Tony described. The way that has sort of, for me at least, captured what his role is in our country is Tony is America's scientist-in-chief. When it comes to issues of science, when it comes to the most pressing issues that we struggle with as a country, when we think about who is our leading scientist. Now, he's got official titles. He's the head of the NIAID. The Web of Science says he's the 11th most cited scholar ever out of 1.9 million scholars that they track. So all sorts of extraordinary accomplishments. But the way I think about it is in the middle of the most difficult moments where we really need to think about who is a scientist we trust, whose science is impeccable and yet whose voice is really captures the mood of the country. There is nobody better. So when we were pulling this event together, the person who was at the top of our list for a keynote, of course, was Tony Fauci. And Dr. Fauci, we are so thrilled to have you here. Thank you so much.
Thank you so much for that really very kind introduction. It's really a great pleasure to be with you here this morning. I'm sorry that I was unable to be earlier on, but there are other infectious diseases around They don't occur necessarily in outbreaks, but I was at the UN General Assembly on tuberculosis for the past few days, which is sort of an interesting discussion that we'll have some other time. So what I'm going to do today is discuss the concept of pandemic preparedness. And by pandemic, I mean not necessarily just influenza, but all outbreak preparedness. And I'm dividing it up into three components that I will discuss relatively proportionately. As you see here from previous experiences, the multifactorial nature of how you prepare, and vaccine development, which is at the core of so much of our preparedness. So let's start off with previous experiences. So I'm going to take the liberty, I was discussing this with Mike Osterholm just a few minutes ago, of the fact that for better or for worse, I have been doing this for a really long period of time. In fact, I was appointed as the director of NIAID 34 years ago. I was 11 years old at the time, and it was sort of a little bit unusual. It was, got people shook up. But one of the things that one has to do in this position is be responsible to the administration, the Congress, and the American people in the arena of infectious diseases. So right about when I was appointed in 1984, I began a series of things that, I don't know if you'd say for better or for worse, but testifying before the Congress. And I'm going to give you a statistic that seems exaggerated, but it really isn't. But I'm telling it to you because it really underscores the importance of infectious diseases and outbreaks and how the country and the world is interested in it. I began testifying before the Congress in 1984, and I have the distinction, positive or negative, of having testified before Congress more than anyone else in history. And that's the truth. So, and the reason is, every time there's an outbreak, there's at least 10 congressional hearings. So if you say 10 times 34 years, you're talking about anywhere between 280 and 300 times testifying before the Congress. So the first time I did, I like maps. And I know I've shown this several times in the past, but I just want to, for those of you who have not seen this, I drew a map, or else my graphics guy drew a map of the United States, and the first time I testified was in 1984. And as you remember, that was the very early years of the HIV/AIDS pandemic. So I put this on the map, and I said, there are outbreaks, and I'm going to come to you to predict for sure we'll continue to have outbreaks. And every year I testify before the Congress. The last time for our budget, just in the spring, I show the same map, but they never get tired of that map because every year I put either 1 or 2 or 3 new infectious diseases. The last time I did this, which was in April or May of this year, 2018, this is what the map looked like. Some of these are curiosities, little blips in the radar screen, and some of them are in great import. Obviously, HIV/AIDS is one of the most important of these, but there are others, such as the pandemic of 2009, such as the Ebola outbreak, and Zika, and chikungunya, and others. So rather than go through each and every one of these, I want to draw on the first part of the talk, previous experiences, and give you some lessons learned before we do a deep dive into how we respond. Because there are different lessons with different outbreaks. There's some common denominators, but there's always a little bit different that you learn. So if you were to take some recent events in emerging infectious diseases, I don't have time to go into each and every one of these, but I want to pick out a few and just drop a couple of pearls about things that we need to remember. So the first one that we'll talk about is SARS, which is an amazing epidemiological phenomenon that we all need to remember in the history of how outbreaks occur. Of course, it has to do with everything from transparency, which, as you know, I see Mike smiling because we went through this together, is the outbreak in the Guangdong province of China, which was sort of sold in the early months to be kind of an unusual influenza. Well, it wasn't influenza. It was a brand new disease In November, the first cases were seen in China. They reported the cases in February, so 3-4 months later, and only when one of the people who was infected went from Guangdong to Hong Kong and stayed in the now famous or infamous Metropole Hotel and infected about 19 people and did what most people who go to Hong Kong do, is they travel to other parts of the world. So 1, transparency, 2, geography, 3, transportation, because as soon as they got on the planes, what they did is that they created, predictably, a global pandemic. Now, that's the bad news. The somewhat encouraging news, another lesson, is don't, in our world of technology, sell short 19th and 20th century public health measures of identification, isolation, quarantine, because that actually, before we could do anything from a medical standpoint, essentially shut down the outbreak. Interestingly, we started and developed a vaccine for SARS. It tells you one lesson that I'm going to get back to over the next several minutes a few times. This was the first dipping our toe into the waters of 21st century technologies, of being able to sequence a virus and get it into a vaccine trial more quickly than you'd ever imagine. Namely, not necessarily having to grow a virus and activate it. And we did this with some DNA vaccine technology. It's interesting, we never had to use the vaccine, and no company was interested in taking it from us and making it into a vaccine, understandably, because this was not economically sound for a company to do that. But as I mentioned, fortunately, and the lesson is that the outbreak essentially burned itself out with the help of typical, well-tried public health measures. Next, H1N1 pandemic, the first pandemic of the 21st century. Now, we all say, those of us who are involved in influenza, that the only thing predictable about influenza is that it's unpredictable. And as a matter of fact, the concept of unpredictability really showed itself because everybody was focusing on the Far East and China. Of course, that's where all the pandemics arise until they don't. So what happened back then? We had the pandemic of 2009. This was really an interesting experience for all of us, but for me it was a particularly interesting experience because what happened is that while we were looking towards China and the Far East, the outbreak actually occurred right in our own hemisphere. I have both California and Mexico down because the Californians say it started in Mexico, the Mexicans say it started in California. It's sort of kind of like the Spanish flu in Kansas or wherever. But as it turned out, it was there. Now, the interesting thing about this, why, I like it as an infectious disease person. I don't mean like to see these things happen, but it's such a fascinating phenomenon. As you all know, in influenza seasons, predictably, if there's one thing that's predictable in influenza, is that the season goes up, it peaks in January, February, comes down in March. In March, we started to see a new virus. We all knew then that it actually was going to be a pandemic. It was brand new, and it was occurring at the end of the season. And the question was, would it spread? I began testifying before Congress saying, absolutely it will. And sure enough, the next month, in April, it was all over the world. And it wasn't the flu season in the Northern Hemisphere. It was April. So what did we do? And this was interesting. So this is a picture of Ann Schuchat, who was acting CDC director at the time, and I testifying before the United States Congress in April of 2009, saying we're pretty sure there's going to be a pandemic. And even though we have this antiquated way of needing to grow virus mostly in eggs, which takes about 6 or 7 months, we're on it. I wasn't so bold as to say we're on it. I was a little bit more modest, but I said, I think we can do this. So it's April, so 6 or 7 months. April, May, June, July, August, September, October. By October, we'll probably have the vaccine ready to go. We know that flu peaks in December, January, and February, so we'll probably get most people vaccinated before we have the peak of the epidemic. I was testifying before the Appropriations Committee. For those of you who follow things in Congress, that's the good committee. That's the committee that gives you money. You usually don't get into trouble with an Appropriations Committee. But then what happened? The children came back to school. And when they came back to school, we didn't have a peak in January and February. So this was the slide in blue of the influenza attack, and in red was the availability of the vaccine. And as I always say, what's wrong with this slide is that the blue line is where the red line should be, and the red line is where the blue line should be. Namely, by the time with all of the little mishaps that always happen when you have to grow a virus as opposed to the modern technologies we have, the epidemic peaked before we even had virus to distribute. So we did the tried and true way of responding, which is the reason why, as I know many of you know and are involved in this, why we're now very bullish on getting alternative ways to develop a flu vaccine. But I show this picture a little bit tongue-in-cheek, because when that happened, here now you see Tom Frieden and I and Jesse Goodman, not before the Appropriations Committee, but before the Oversight and Government Reform Committee, trying to explain to them why we knew about this 7 months ago, and we still had a situation where we were woefully behind in getting vaccine to the American public, which really triggered, and I'm not going to spend much time on it, something that we have been very, very much involved with, and that is trying to develop a universal influenza vaccine. We developed a strategic plan. We could talk about it during the question period, but it's something that I feel we really need committed to do. It's not going to happen overnight. It's not going to be one single situation. It will be an iterative process that will ultimately get there. Okay, next is Ebola in West Africa. As you all know, Ebola was discovered in 1976. We've had 24 or so outbreaks over the years, ranging from anywhere from 2 or 3 people to a couple of hundred people, mostly in Central and Southern African countries. Then came the 2014-16 outbreak. Some lessons from previous experiences. One, transparency and ability to communicate. The first case was in December of 2013. It wasn't until March of 2014 that the world woke up and realized that we really have a problem here. And what happened, again, I don't need to tell the audience this story because it's well known. We had a catastrophic outbreak in 28,000 cases, 11,000 deaths, in what we call the perfect storm of infectious diseases. And every once in a while you get a perfect storm, and I'll get back to that in a minute. Namely, something that generally would not happen unless you had multiple factors. 3 countries, porous borders, population totally distrustful of authority, and no healthcare delivery system. And that's what happened. What are some of the brighter sides of that, of lessons learned? And that is, you can do ethically sound and scientifically sound research in the setting of an outbreak, and you can learn things, and we did. Cliff Lane and my group started the Prevail in Liberia, the Partners for Research on Ebola Virus in Liberia, partnered with Musaka Fala, who's here in the audience, to doing some clinical trials in vaccines and therapeutics and long-term follow-up in survivor studies. We analyzed this, and the lesson we learned, published by the National Academy, was that you can actually do sound research in the setting of an outbreak. But I want to tell you something else of a lesson we learned is about the unrealistic fear sometimes we have with outbreaks in this country. And that was manifested when Ebola came here. When we had an individual, Mr. Duncan, who went from Monrovia to Dallas, was not aware that he was infected, though he was aware that he was exposed, went into an emergency room, left, came back days later, and exposed 2 nurses who in the first few days of taking care of him did not realize he had Ebola and were not completely protected with total body personal protective equipment. And what happened is that 2 of the nurses got infected, Amber Vincent and Nina Pham. And what happened is that all of a sudden, the attention of the United States turned from great concern about what was going on in West Africa to almost forgetting about West Africa and focusing on something that was not going to happen in the United States. We were not going to have a massive outbreak because that's not the way Ebola spreads. And in fact, USA said it very well, that fear spreads faster than Ebola, and that's for sure. But we learned something from that because in the United States, multiple years of training, certainly at the NIH, but also in Nebraska and Emory, of a special Ebola treatment facilities. We had been working on that for years, even before Ebola, because we were the area that would be taking care of people, particularly at Fort Detrick in Maryland, who might have accidentally gotten infected with something. So we needed a BSL-4 intensive care, and we did that. So we took care of Nina Pham, and here's a picture that was went viral. It was on the front page of the Washington Post, of when we discharged Nina after taking care of her for about 2 weeks. And the reason I was very adamant about having a picture with my arm around her is that the other lesson you learned in Outbreak is stigma. Not only were people who were infected with Ebola stigmatized even though they got better, but the people who were taking care of Ebola were stigmatized. I don't like to say this, but it happened when we started taking care of Ebola patients at the NIH Clinical Center. The children of the nurses who were helping us were stigmatized against in daycare centers in the United States of America, right outside of Washington, D.C. One last thing about Ebola before I go on. We also then had a very interesting experience that has to do with the medical care of Ebola patients. One of Paul Farmer's physician's assistants from Sierra Leone got infected. Paul wanted the patient to be taken care of me and my team, so we arranged to air evac the person. And here's a picture of 3 members of my team getting ready to receive the patient on the back door of the NIH Clinical Center. And here we are bringing the patient up. And we had a system in the NIH Special Clinical Studies Unit, a 24-hour system in which you always have one infectious disease person and one intensive care person there with all the Watsons and the nurses getting ready to go. I did sort of a little mini sabbatical for about 4 weeks to join the team. I didn't want the people on my team to do this if I wasn't going to do it. So here we are. This is Dan Chertow and I getting ready. This is taken at 2:45 a.m., getting ready for our 2-hour shift. And this is Dan getting ready to go in to take care of this patient who will remain anonymous. And this essentially happens every 2 hours. So here I am putting my stuff on. I always give credit to this lady, this woman, this experienced nurse here behind me, and the individuals here who are checking us out. You can't have this type of an approach without a very experienced team who are responsible not only for the care of the patient, but for your safety. And here is Dan and I taking care of this patient that we reported in the Annals of Internal Medicine, who was one of the sickest patients I've ever seen in my life. But because of intensive care, Ebola is not 70, 80, 90% fatal. If you can maintain the person in a modern intensive care, the mortality is significantly lower. I can't say what it is, except that only one patient who was taken care of in the United States died, and that patient was dramatically ill even before they got into the intensive care. So it's much lower. And here again, I show the person was admitted, walked out of the hospital. I still see the patient in my clinic, comes in about every 6 months, doing very well. Moving on quickly, Zika in the Americas. We all know the story. It was discovered in 47, first patient in 52, 2 outbreaks in the Pacific Islands. We didn't pay much attention to it. Why? It was thought to be a trivial disease. 80% of the people had no symptoms. You go back here and you say, well, wait a minute. It was 2013 in French Polynesia. Why weren't you developing a vaccine then? Another area of a perfect storm. What happened is that when it finally got to Brazil, and the N was high, as shown by this green line, then we found out something we would not have predicted, that it wasn't inconsequential at all for pregnant women. And that's when we had the disaster of the microcephaly, which was really the tip of the iceberg, because there were many other things associated with this particular involvement of the central nervous system. So I'll stop there with previous experience. Let's move on quickly to how multifactorial pandemic preparedness is. And that's what we'll talk about. So here is some of the optimum preparedness factors. We obviously don't have time to go in through all of them. It's surveillance, laboratory capacity, communication, all the things we talk about. Since I'm from the NIH and I want to talk to you a little bit about the research response, let's just focus on that for a moment. The critical role of biomedical research in pandemic preparedness, you can break it down into 3 major components. First, the basic research portfolio. You can do research and you must be asking questions as outbreaks are evolving. You can't only do it in a post-mortem, and that's exactly what we have been doing. We've done it with, when I say we, I mean the community, us. We do it with Ebola, we do it with Zika, we do it with chikungunya, we do it all. You can't do that unless you have some capacity, both domestic and international, and that's in fact what we do have. We have vaccine and treatment evaluation units in the United States. We have the Centers of Excellence for Influenza Research and Surveillance, CERS, globally throughout the world. And we have intramural international collaborations. So basic research portfolio, research capacity, and then the bottom line of it all is, what do you do about it? And that is countermeasure development. The United States Public Health Service has that. It's called FEMC, the Public Health Emergency Countermeasures Enterprise, of which the NIH is one of many partners, including the CDC, the FDA, ASPR. So I'm going to tell you a little bit about the NIH approach to this. As you all know, I'm sure, we do everything from basic to clinical research, research capacity, with the ultimate thought in mind of developing countermeasures. I have therapeutics, diagnostics, and vaccines. But since we don't have a lot of time, let's just focus on the issue of vaccines. So vaccine development for pandemic preparedness, we've thought about it a lot. I've mentioned to you a concern that we all have about having to grow a particular pathogen, usually a virus, either inactivate it or attenuate it, make it a vaccine. It takes a lot of time. So all of us in the field have been figuring out how we can have the 21st century approach towards vaccine development to prepare for any outbreak, influenza or anything else. So I've divided that up into 3 components shown here on this slide. They overlap, they interdigitate, and they complement each other. The first is the issue of priority pathogens. Anticipate what might be something that likely would be an outbreak. That has some positive aspects to it and some negative aspects. If you're right, you're ahead of the game. If you're wrong, you may have wasted time. But there is a positive aspect to it. So the WHO has this R&D blueprint which aims to reduce the time between when you declare a public health emergency and you have a vaccine ready. So what they've done is that they've named a number of diseases to be urgently addressed under the R&D blueprint. This chart showing as of February 2018, and you see them listed there. This actually overlaps and is complements with a relatively new organization, CEPI, which is the Coalition for Epidemic Preparedness and Innovations, which are working with a number of partners, including us, to develop vaccines against priority pathogens. And these are the first 3 of the priority pathogens that they have chosen: MERS, Nipah, and Lassa. Not a bad choice of 3. But they're also doing something, and this is something that we at the NIH feel strongly should be the major direction of what we do, and that is to develop platforms, to perfect platforms that are easily interdigitated with whatever outbreak you have. So here are some of the vaccine platform technologies. And on the right is examples of diseases in which we've used them. So genetic immunizations, DNA and RNA vaccines, as you can see on the right, we started off with SARS and we went through. Viral vectors, recombinant proteins, nanoparticles will be key in developing a universal flu vaccine, for sure, as well as virus-like particles. Now, let me tell you why it's important to perfect a platform, because you get better and better at it, and the FDA gets more comfortable and more comfortable with it. So let me give you an example. As I mentioned, the first one we did was SARS, and you're all familiar with the DNA vaccine, probably going to be replaced by mRNA soon, but the DNA vaccine, where you stick a gene that encodes for the relevant protein into a plasmid, you inject it into an individual, they make viral-like particles. But this next slide is really interesting, because I mentioned to you that when we did this with SARS, at the time, back then, it was the quickest from sequence availability to a phase 1 trial. But each time we did it, it got better and better. It took 20 months from the time we got the sequence of SARS to put it into a first-in-human trial. We also did it with the H5N1 2005 pre-pandemic. We got it down to 11 months. We tried it again with H1N1, and from sequence to phase 1, it was 4 months. With Zika, we now have the indoor record, because from the time we sequenced Zika to get it into a trial, it was less than 4 months. And that trial is ongoing now in South America, accruing individuals, and we're getting a lot of safety and immunogenicity data, even though there aren't a lot of cases of Zika. And the FDA has been extremely cooperative and encouraging in us that we might be able to actually get approval purely based on data from safety, immunogenicity, and bridging to animals. The final one is the prototype pathogen approach, something that has been pushed by a number of people, particularly Barney Graham in our vaccine research center at NIAID. And that is to pick out a particular genus like flavivirus and look at all of the pathogens that are in that group. Japanese encephalitis, West Nile virus, Zika, dengue, yellow fever, things that we've had to deal with. There are some commonalities there. And what we're doing now is we're trying to apply common denominator strategies and tools from experience with one virus to inform vaccine design for related viruses, so that we don't have to keep reinventing the wheel every time we do something. So there's basic virology that has commonalities. Assays are similar. Animal models can be quite similar. Antigenic targets are similar. The platforms, if you have a DNA platform that works for West Nile, it's going to work for another one in that genus, for sure. I mean, you have to prove it always, but it is very unlikely that there's going to be major differences. The potential immune correlates are likely similar if you're making a response against a particular, in this case, a flavivirus, and the manufacturing is similar. So, in order to stay exactly on time, so let me close up with just a reiteration of what I've said. that pandemic preparedness is something, obviously, that we need to take very seriously. It's what we're all about in Outbreak Week. And there are things that we need to continually draw on. We continually learn from previous experience. Lessons learned are very important, and they are continual with every outbreak. We need to understand and utilize the multifactorial nature of pandemic preparedness. It's not unidimensional. It's not only science, it's not only surveillance, it's not only communication. It involves multiple agencies, public and private sector. And then vaccine development is critical, and that's the reason why we have to keep improving on it. And I'll end with a slide that I always end with, and it relates to something that many of us, as I look at my colleagues in the audience, have been talking about for some time, is what I used as the title here, Emerging Infections, a Perpetual Challenge. And it really is. If emerging infectious diseases are a perpetual challenge, then we, as the responders, need to be perpetually prepared and perpetually able to respond. Thank you.
So we have about 10, 15 minutes, I think, for questions.
Yes, of
and
course.
So people could come up here, and I'm going to ask the first, if that's okay, which is, my question is, I stole this from Mike Osterholm. It's about missiles, which is, we don't think a lot about the business model for missiles. There's a very specific business model, which is the government buys them, doesn't use them very often, but feels it's really important to have them for national security.
Right.
That's not how we think about vaccines for pandemics. The government, NIH, CDC, USAID play very helpful roles in important parts of that process. But then we leave that last part up to the market, kind of, sort of, but we know that that doesn't quite work. So the question is, why don't we treat vaccines for pandemics the way we treat missiles, or should we, or is that a bad analogy?
No, it's a reasonable analogy, and it's something that we really have struggled with for years and years. I know, I'm certain that Mike tickled you with that, because we've been discussing that for so long. There is a feeling, probably based on some reality, that the federal government is not a vaccine production organization. And the expertise of being able to do that and say we don't have to worry about a company getting involved, let's do that, is probably not feasible for keeping it warm and going. Having said that, I do believe that we should take a bit more responsibility in developing a vaccine without necessarily always relying on the pharmaceutical companies. We tiptoed into that a bit when we made hundreds of millions of dollars of investments to develop a plant in North Carolina to make the switch from egg-based to cell-based. You know, that was, I think, a good thing, but it wasn't